We can make it from tryptophan, but if you don't make it from tryptophan, you have to get it either from niacin or from your diet. Tetrahydrofolate is another, actually this is used in methyl group transfers. There's basically, right here in this molecule ends up being able to carry a methyl group. So this is actually very important in nucleotide metabolism. We'll talk about that again in a second lesson. Actually, there's a story about a physician from Great Britain who went to India to try to find out the cause of pernicious anemia, which was being found particularly in women, pregnant women working in textile factories in India. Basically, when you're pregnant, because the fetus is developing, there is a lot of DNA synthesis. Those cells are rapidly dividing into nucleotide synthesis to be able to allow those cells to replicate and grow. And nucleotide, to make DNA, to make a nucleotide, requires a lot of this particular cofactor to be able to add methyl groups to the pathway. He's the one that recognized it was a dietary factor that was missing, and basically cured it by having people eat yeast extract, Marmite. If you guys know what's there, Marmite. There's another one called. Anybody have any Marmite? You go to like some specialty bread shops here, you can get it's vile stuff, I tell you. What's that? Vegemite is the other one. Vegemite, yeah. Vegemite and Marmite, basically the same thing. It's a yeast extract. It's kind of salty. I don't know how to taste it. It's an acquired taste, basically. You use it like peanut butter or something, put it on toast and that sort of stuff. Basically, it's nutritionally it's very, you want to have a lot of folates in there. So, basically, by feeding people the Marmite and Vegemite and yeast extract, we were able to work out thiamine pyrophosphate, this is another one. This is a very important cofactor in aerobic metabolism and the pyruvate decarboxylase reaction. Again, it's also vitamin B1 cofactor. Here's a big fancy one, cobalamin, vitamin B12. And what this basically does is well, it provides this cobalt function here. And if you go through and you see this cobalt here is coordinated in a particular way. This is also important in nucleotide metabolism. It's important in, you know how DNA lacks that hydroxyl group, and the 2' hydroxyl group, and we talked about how important that hydroxyl group, that lack of that hydroxyl group was in terms of the overall stability of the DNA molecule. Well, the enzyme that actually removes that hydroxyl group requires this particular cofactor in here. When you think about it, that's kind of significant because from an evolutionary life standpoint, it's generally thought that for a long time there was an RNA-only world. The DNA didn't exist, and then at some point in time, the DNA as a nucleic acid was evolved. Well, what do you need to make DNA? You need to be able to remove that hydroxyl group from that. And then when you have this sort of structure that's being involved, you recognize that that's not a particularly easy reaction just to have happen. And so, from an evolutionary biology standpoint, it's kind of interesting how that particular reaction evolved, and particularly when you start noticing that it requires such a complex cofactor in there. Yeah. So anyway, so those are some examples of some enzyme cofactors. I don't know what percentage of enzymes require cofactor. My guess is like maybe 10%, something like that. Most of them do fine with the 20 amino acids, but a significant number of them don't. All right, where are we? I think that's where we left off. The next thing I want to talk about is a little bit more of a theoretical aspect. We're going to go in and start talking about specific catalytic mechanisms. Before getting into that, I want to sort of create a theoretical framework, and we're going to come back to this when we look at enzyme kinetics as well. Just to remind ourselves, when we have a chemical reaction, what I'm talking about here is activation energy and catalysis. Let's take a chemical reaction. We're going to do the famous chemical reaction A goes to B. And if we draw a chemical, an energy diagram, and on this axis here, we've got free energy, G. Again, we haven't looked at this quantitatively, but this is a reminder, free energy, there's two components. There's the entropic component, and then there's the enthalpic component, delta H and delta S. So this basically brings those together. And if we have A, might have a particular free energy, and maybe our B might have another free energy. Doesn't necessarily have to be lower, but I'm going to put it lower. And then if we look at the difference between these two, this is delta G, the change in free energy. So when the free energy of B is lower than A, is this reaction spontaneous? Yeah, that's right. It's spontaneous, yeah. Let's just kind of on the side, if A was up here and B was down here, is the reaction more spontaneous? Yeah. If A is up here and B is down here, is it faster than if A was here, down here? Not necessarily. The delta G tells you whether or not it's spontaneous and how spontaneous, but doesn't tell you anything about the kinetics. It doesn't tell you how fast the reaction is going. What tells you how fast, because we have to see what our path is. And our path is that we have a transition state up here, right? Often marked like this. This is the transition state. So our path going from A to B is going to look something like this. And this distance here is known as delta G star, which is known as the activation energy. And it's the size of that activation energy that actually controls how fast the reaction goes. Now, one of the things I always like to do is think about, okay, why? You know, what is it really that's going on there? Why is it that activation energy controlling how fast it is? What sort of, let's look at the detail. And basically, what you do is you look at we say the energy of A is here. But that doesn't mean that, you say we have 1 mole of A, right? 6.022 times 10 to the 23rd molecules of A. That doesn't mean every single molecule has exactly that energy, but that there's a distribution of energies, and what is it? Basically the average is A. Okay? some of those molecules have a little bit more energy. Some of those molecules have less energy. Anybody know the name of that distribution of energies? Anybody in 142? We've gotten it down here. Boltzmann distribution. It's the Boltzmann distribution of energy. Okay, so there's a distribution of energies that center around A. That means some of them are a little higher, some of them are a little bit lower. Okay. Now, a few of them, maybe just a very few of them, actually have high enough energies as a transition state here. Those are the ones that will actually move forward. Okay, so let's say that we've got say we've got 1,000 molecules here. Okay, 1,000 molecules here, and 10 of those 1,000 molecules in a minute's amount of time, 10 of those molecules actually have enough energy up here. So in that minute, 10 molecules will actually make their way over. And in the next minute, we've got 990, so almost another 10 will go over. And then basically, it slowly goes over, right, as that fraction of molecules that have that energy reach that state. Now, if my transition state was down here, the lower energy, then a much higher fraction of molecules would actually have that molecule. Maybe 20 or 30% of the molecules would actually have that energy. And so now you have 20 or 30 molecules every minute going over rather than just 10 up here. So the reason that the activation energy controls the rate of the reaction is because it basically describes the percentage of your reacting molecules that are actually going to have enough energy to be able to react. And the higher the activation energy, the lower that percentage is, so thus the whole reaction will go slower. Anyway, so that's the kind of the detail behind that. All right. Now, so what does a catalyst do? A catalyst enhances the rate of a reaction. Okay, it makes it go faster, right? Doesn't change the free energy at all. It just makes it go faster. So that means down here, there must be, if we're making it go faster, we must lower the energy of the transition state. So here might be the transition state for the catalyzed reaction. This is the uncatalyzed reaction up here. So somehow the catalyst is able to lower the energy of this, and so now this difference in energy here is the delta G of the catalyzed reaction. This distance in between here, the amount that is lowered, basically gets at how much the power, the catalytic power of the catalyst, how much power. And this represents it's known as the delta G star, which equals the delta G star minus the delta G of the catalyzed reaction. Here, I'll maybe make it a little clearer. This is uncatalyzed minus the catalyzed. Now we can actually talk about the rate enhancement. We can quantify the rate enhancement to the delta G divided by RT. And, a rate enhancement and enzyme catalyzed reactions are, I think I said any place from like 100 to a billion fold. You say a million, and a million might be typical, okay? So if we have a million fold rate enhancement, so if we, that would be 10 to the 6. This, I'm just going to say that it ends up being 34 kilojoules per mole. This is at 25 degrees Celsius. So that kind of gives you an idea. So basically, whatever catalytic mechanism we've got to provide 34 kilojoules of stability of the transition state to see the type of rate enhancements that we typically see in enzyme catalyzed reactions. So we're going to go through and we're going to give examples of different types of enzyme catalysis, and basically they all have to add up. Each of these different types of enzyme catalysis are going to contribute a little bit to this overall value of 34 kilojoules per mole, more or less, of stability of the transition state. All right. So now we're going to get into the meat of it, catalytic mechanisms. And different textbooks will have slight variations on these categories I'm going to give you. The one I'll give you are probably pretty standard. And again, you got to think of the broad thing. What this is trying to understand how enzymes catalyze reactions, basically is trying to understand how life happens. It's basically life is a bag full of catalysts. All right. So the first mechanism we're going to talk about is acid-base catalysis. And here, the enzyme actually provides a proton where it's needed or accepts a proton where it's needed. And the example I'll provide is actually catalyzing a reaction that we already talked about a little bit when we talked about RNA stability. And this is ribonuclease. This is an enzyme that takes an RNA molecule and hydrolyzes the RNA molecule, breaks it up. So I'm going to start my mechanism here. I have plenty of room. All right, I'm drawing an RNA molecule. Good review here. you remember we talked about RNA stability, we talked about the role of this hydroxyl group here, this 2' hydroxyl group, and the fact that it can actually do a nucleophilic attack on this phosphorus to ultimately result in the hydrolysis and cleavage of the RNA molecule. And that was one of the big advantages of DNA. By removing that hydroxyl group, you eliminate the possibility of that reaction. Nonetheless, there's a lot of enzymes around that actually degrade RNA. Now, why are there enzymes around that degrade RNA? Well, one, if you're a bacteria, it's any organism really, it's an antivirus mechanism. There's a lot of viruses that are RNA viruses, so you have ribonucleases to basically deplete those viruses. And then also just during the normal turnover of RNA and all that stuff, you might have some ribonucleases. So the way a ribonuclease works, okay, is to try to promote this this nucleophilic attack of this oxygen on that phosphorus. The way it does that is by positioning a chemical imidazole ring from histidine residue. So I'm going to put one here. You guys, if I draw this, you guys have to police me a little bit. If I start drawing a pen of the orange part there, always a bit of an adventure. All right, so say we have that nitrogen. Here's our histidine. And I'm just going to put enzyme here for the correct line. So you basically have to recognize this is the side chain of a histidine residue here. We've got a pair of electrons there. And then there's another one that basically sits over here. And this one is actually in the protonated state. Now what's going to by having the enzyme basically fold around and position these two imidazole rings from histidine side chains right at the right spot, it allows for this to happen. Basically, this pair of electrons can come down here. Okay, that allows this pair of electrons to come over and attack that phosphorus, okay, and then this allows this pair of electrons to come down here.
Are those first electrons moving to the oxygen?
No, to the hydrogen. To the hydrogen. So then we get this intermediate state, basically that looks like this. and we'll talk about what happens to it. All right. So let's look at basically what happened is, so if we move this bond down here, now we have this strand, now this is able to actually go away here. We made this intermediate, this cyclic phosphate intermediate here. Let's look what's happened. We've gone to one imidazole in the protonated state, another imidazole in the deprotonated state, and now we basically swapped roles. Now this is in the deprotonated state, and this is in the protonated state. So what the enzyme is receiving a proton here, and it's donating a proton here. So again, this is the acid-base catalysis part. It's a donation it's the donation and the reception of the proton. Okay, now this reaction isn't totally over because we don't leave this RNA molecule in this cyclic position, but rather we have a water come in, and so then the water comes in and comes in right here, and we can basically now we can actually do everything backwards. Basically now this can come up here, and then this can come back here, and these electrons can come back onto the nitrogen, like so. Back to our basically go back to our original state of the imidazole rings, where this is deprotonated here, this one is protonated over here. Boom. Okay, so that's kind of cool, right? Boom, 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 boom. And so basically the whole, I don't know how long a ribonuclease is. It's not that big. This might be 30 kilodaltons, maybe 300 amino acids. And the whole idea is those 300 amino acids all fold up to get that secondary, tertiary structure wrap around, and really everything that they're doing is basically to position two imidazoles right at the right spot, okay? As well as finding a place for the RNA to bind and position these right at the right spot, and that allows those imidazoles to serve as either both an acid and the base of the reaction, and it greatly accelerates the rate of the reaction. Now, one of the outcomes of this is that if you look at that, what's the pK of an imidazole ring? 6.1? 6.1, yeah, 6.1, something like that. So when you think about this, in order for this whole reaction mechanism, you've got to have an environment basically where these imidazoles can be in both the protonated and deprotonated state, right? They have to be going back and forth. So if we try to do this reaction at pH 2, okay, then both of these guys would be in the protonated state, and they wouldn't really help much. Similarly, if we did it at pH 10, both of them would be in the deprotonated state, they wouldn't help much. But if we're at a pH 6, more or less, okay, well then they can actually be in pretty much in either state. So if we look at, and this is often done, is if we look at the activity of an enzyme relative to pH, activity meaning the catalytic rate of the enzyme, that you'll get a curve in which there'll be an optimum pH. And often, if an enzyme is involved in acid-base catalysis, that pH optimum is going to be right at this pKa of the side chain involved in the catalysis. And so this is going to be the pKa of the imidazole histidine. And that was, in the early days, that was one of the ways of trying to guess what residues might actually be involved in catalysis. If you had an enzyme that had a pH of around 4, pH optimum around 4, and you think it might be involved in acid-base catalysis, where you say, well, maybe one of the carboxyl groups from aspartate glutamate are involved. If it's around 6, well, histidine. If it's up a little higher, if it's up around 8 to 9, maybe the histidine might be involved, something like that. So by basically looking at the pH curve, you can get an idea of what side chains might actually be involved in the reaction. Here's another kind of sort of tidbit for those of you who are actually involved in the lab sort of stuff. So one of the hardest things to do in biochemistry and molecular biology is to isolate RNA well. The problem is because in the environment there are so many ribonucleases because organisms make them to protect themselves against RNA binding. So on your hands, I can tell you there's going to be a whole bunch. So if you take your hand and dip it into an RNA solution, the ribonucleases from your hands are going to get in there and going to start chewing up your RNA. And so get a really high quality RNA prep, you have to have somebody who's really fastidious, right? Really anal, basically, keeping everything super clean. One of the things you do is to try to inactivate any ribonucleases is you treat all your glassware with a molecule called diethyl pyrocarbonate, or DPC it's called. What DPC does is it goes in and it chemically modifies the imidazole rings. Okay? And so basically by treating all your glassware and your buffers and the water that you use in buffers and everything with diethyl pyrocarbonate, you serve to inactivate any of the ribonucleases in there and will allow you to get really high quality RNA. All right. Let's do one more. Covalent catalysis. I don't like to rush through these. I know they're pretty cool. It really kind of gets into chemistry. Again, it's gets at the fundamental question, very fundamental question. Next is covalent catalysis. So here we're going to provide a pathway that doesn't exist in the uncatalyzed pathway that involves a covalent enzyme substrate intermediate. By the way, while we're going through these enzymes, we should also be thinking, remember we're classifying enzymes? All these different enzymes, what were the lyases, the oxidoreductases, the hydrolases, I'm trying to remember all that. I gotta memorize it before the next exam. Anyway, so what about this one? What class? After that one, the ribonuclease. What's that? Hydrolase, right? Here it's all in the water. We're basically using the water to break up the molecule. Hydrolase. All right. So here's an example. The example I'm going to give for covalent catalysis is acetoacetate carboxylase. So acetoacetate this molecule here what can basically happen is these electrons can come down here, these electrons can go over here, and these electrons come up here. This is in the uncatalyzed reaction. What comes off is carbon dioxide. Then along comes a proton, and I should mention one of the fun things about biochemistry is a lot of these reactions will give off a proton and take in a proton, and you just kind of go with it. You don't necessarily, I don't worry too much about balancing protons or anything like that. But, don't think, where that proton? Protons out there. It's pH 7. There can be protons out there. Pick one up here. And then basically what happens here is that then this comes back down here, one of these guys comes back down there, and you end up making acetone. This is part of ketone body metabolism, actually. Ketone bodies are a class of molecules that become important energy sources when you get really low blood sugar. So you see it in people that are either fasting, when your blood sugar goes way down, or people with diabetes who their blood sugar is unregulated, so it's peaking and going in valleys. So one of the tests is for ketone bodies. There's a sign of diabetes. What else? Ketone bodies. Oh, and then if you're starving to death. All right. So let's go ahead and do the enzyme catalyzed reaction. The enzyme catalyzed reaction is going to have an amino group from a lysine side chain positioned just in the right place. And that allows for the formation of a shift base what comes off here is, actually, let me do that in red. Form that shift base there. So this is a species that basically we don't see in the enzyme catalyzed reaction. Now this is basically able to handle the decarboxylation at a much lower activation energy. So we basically go through a similar kind of thing. And with this is going to come over here. So these electrons are coming up there. You get our carbon dioxide coming off. And then, I'll tell you what, I know this is an awkward place to stop, but we're going to go ahead and stop there. I'm going to bring that up on Monday and we'll kind of go through this, kind of review it.